Hello and welcome back to WIPS Biochemistry once again, and this time we will be discussing chemiosmotic theory or chemiosmotic hypothesis as part of cellular respiration. Well, in one of the earlier lectures, we had discussed that electrons released in the oxidation of NADH and FADH2 flow through the electron transport chain or respiratory assemblies to generate a proton gradient. A proton gradient will be generated across this membrane, inner membrane of mitochondria. And protons jump from like this mitochondrial matrix to intermembrane space as the electrons pass through this assembly. So this is like, yes, electrons. As the electrons pass through, you can see protons jump. Yes, this one and again another one. So this is how protons gets accumulated here in the intermembrane space. And these protons then flow through this structure, F1 complex ATP synthase. It's also called complex 5 also, so as to generate ATP from ADP and inorganic phosphate. So this is what we are going to discuss in this short video. And thus far, what you have discussed in the previous lecture was the flow of electrons from NADH to oxygen, which is an exergonic process. But now what we are going to consider is how this process was coupled to the synthesis of ATP. Now, synthesis of ATP, which is actually an endergonic process. And here. It was in 1961 was Peter Michell, a Nobel laureate, who proposed that electron transport and ATP synthesis are coupled by proton gradient across in the mitochondrial membrane. And in this model, the transfer of electrons through respiratory chain leads to pumping of protons from matrix to cytosolic side of the inner mitochondrial membrane, that is membrane. The space between inner membrane and outer membrane is called intermembrane space. So like this you can see with the protons jumping from matrix to like this. So yeah, the H plus ion concentration becomes lower in matrix as a result, and an electric field with the matrix side negative, this side negative, and this side intermembrane space or the cytosolic side positive is generated. And this Peter Michell's idea is called Chemiosmotic hypothesis. So this is what we describe as chemiosmotic hypothesis. It is that this proton motive force is what drives the synthesis of ATP by ATP synthase. So this is a structure of ATP synthase. It is composed of a proton conducting FO subunit and a catalytic F1 subunit. So the role of the subunits are clear here. This one is FO, which conducts protons from this intermembrane space towards matrix. So this you have to note here, this is the side which is the positive side where there is accumulation of H plus, and this is the matrix side, the matrix of the mitochondria, the matrix side of the mitochondria. Now, this structure, it's a large complex enzyme that looks like a ball on a stick, this 85 Angstrom unit diameter ball, which is called the F1 subunit, it protrudes into the mitochondrial matrix and possesses the catalytic activity of the synthase. Even the isolated F1 subunit is having the synthase activity here. Now this F1 subunit consists of 5 types of polypeptides, as you can see here, 3 alpha subunits, 3 beta subunits, then gamma, delta and epsilon also are part of F1 subunit. The alpha and beta subunits, which make up the bulk of the F1 here, they are arranged alternately in a hexameric ring. The beta subunits are here, which participate directly in catalysis. The central stock consists of this gamma and epsilon subunits, like here. The F4 subunits, on the other hand, is a hydrophobic segment that is embedded in the inner mitochondrial membrane here. The FO contains the proton channel of the complex. Here, the O stands for oligomycin, which specifically inhibits FO subunit. And this channel consists of a ring comprising of 10 to 14 C subunits that are embedded in the membrane. The proton channel depends on both A subunit of FO portion and the C ring. The FO F1 subunit are connected in two ways. One is by central stalk. This is delta. This is gamma and epsilon stalk, and as well as by through the exterior, this beta. So this B dash subunit, and the delta subunit. So there are two connections between these two subunits, as you can see here. And we can think the enzyme as consisting of two functional components also. It is this moving rotor, which is composed of C ring, gamma and epsilon portions, and so this is a rotor which is capable of rotating, whereas the rest of the portions, so these rest of the portions are stationary, and they can be described as stator. So this means that structurally FO-F1 units are like this. The distribution is like this, but functionally it's like this: C, then gamma and epsilon, whereas the rest of the portion, this is the stator portion. Now, how do protons actually get back to mitochondrial matrix? So this is what chemiosmosis explains. So it is by this process. The rotor, which consists of C ring as well as this gamma and epsilon, they are capable of rotating, as we can see in the figure. So when the rotor rotates, what happens is that H plus passes from the intermembrane space, that is P side, which where there is accumulation of H plus ions or protons. The protons pass from intermembrane space through the rotor to the matrix side. So that is what happens, like this and like this. So this is actually this is the energy that gives the energy for phosphorylation of ADP to form ATP. So that is done by ATP synthase, the synthase portion of FO-F1 complex, that is F1 portion. So this is basically what chemiosmosis explains. And this theory is further supported by a wealth of evidences. And indeed, electron transport does generate a proton gradient across the inner mitochondrial membrane. That has been verified. The pH at intermembrane space, the P side, is 1.4 units lower than the matrix side. The pH is lower, which means just having a concentration accumulation of H+. And the membrane potential is 0.14 volt across the inner membrane. So P side is being positive. So that also been measured and verified. So this is one of the evidences to support chemiosmosis. Now, for the addition of acid to the external medium, which actually will reduce the cytosolic side, the P side, the pH of the P side. When acid is added to the external medium, the pH of the cytosolic side goes down. Then this also is found to generate ATP in mitochondria, even in the absence of electron transport. Then further, uncouplers, which disrupt coupling of electron transport and oxidative phosphorylation, as well as certain ionophores, which actually support leakage of H plus ions back to matrix. These agents are found to tend to disrupt ATP synthesis. So these are the evidences which supports chemiosmotic theory by Peter Michell. Now further, there is another theory, binding change mechanism, which was proposed by Paul Boyer. It argues that the actual role of proton gradient is not to form ATP, but to release ATP from this synthase complex. So that is another theory which we will discuss in one of the future lectures.